Good afternoon, and thank you for the opportunity of giving this presentation on the development of biotoxin gene quantitative PCR assays for the monitoring and management of biotoxin risk in aquatic environments. These aquatic environments relate to biotoxins produced by cyanobacteria in freshwater environments from estuaries and rivers, extending into brackish interphase to the marine environment, biotoxins produced primarily by dinoflagellates. And really, why should we be so particularly concerned about better tools and better prediction for biotoxins? And even though we understand the full implication of acute exposure to these biotoxins and manage these risks extensively with the systems we have in place at the moment, the longer term implications that probably drive better research or better understanding of these threats have only recently started to be published or analysed. And in a recent paper from Environmental Health in 2015, there was a study of blooms and their relationship to non-alcoholic liver disease at a county level, looking at county-specific gender and age standardized mortality rates from non-alcoholic liver disease. And they use phycocyanin levels using medium resolution imaging spectrometry, or MERIS, to look at clusters of death from non-alcoholic liver disease. They analysed the relationship between cyanobacteria bloom and death from non-alcoholic liver disease. What they found is that 62% of counties, almost 2,000 of the 3,000 counties, showed signs of cyanobacteria blooms measured by MERIS. By regression analysis, they showed the bloom coverage was significantly related to the risk of non-alcoholic liver disease, and that risk increased by 0.3% for each 1% increase in bloom coverage in the affected county. And this was adjusted for age, gender, education and race. So if we looked at a nominal increase of 5 to 10% of blooms over the next few decades, you would immediately see a tenfold increase in liver disease or death due to non-alcoholic liver disease. And to some degree this should be driving the discussion about the resources necessary for better outcomes or better prediction for the management of these blooms. And in fact, this has been well recognised that there is a need for a reliable early warning predictive process. And research is currently underway in spatial imaging analysis, chemical and physical prediction with sample processing units that are remote. But genetic tools have been proposed, and in fact they've been proposed for a fairly long period of time. At a proceedings of the Interagency International Symposium on Cyanobacterial Harmful Algal Blooms, a number of key objectives or research agenda items were noted, and one of those was to identify and prioritise genetics or omics research needed to improve prediction in cyanobacterial HABs. So this has now been sort of 7 to 8 years since this meeting and this recognition of the need for better tools or prediction for monitoring and measuring harmful algal blooms. So biotoxins are toxic compounds that are biosynthetically produced as secondary metabolites with little understanding to why and specifically when they may occur.
So current testing looks at morphological identification and counting, and the detection of the toxin is analytically or immunologically performed. And species ID can be sometimes very ambiguous, and certainly counting can be inconsistent, has a lot of problems with reproducibility. And so the challenge of cyanobacteria monitoring is identifying the cyanobacteria, determining if it's toxic or non-toxic, or what's the potential of it becoming toxic to a level that's of immediate health risk. And I've got to sort of say, this is a picture of the Sydney's water source back in 2007, when it was down to 27% of its capacity, and massive microcystin bloom hit, and this drinking water is for 4.5 million people. And I'll reflex back to this particular event later on in the talk, but it was certainly a significant event. This is, I use this as an example. This were the pre-2010 guidelines for alert levels for the Australian guidelines for management of cyanobacteria. And they dealt with levels 1, 2, 3, 3A, 3B as far as, and they looked at total counts of specific toxic cells. And at alert level 2, you would actually look at, you would assume it was potentially hazardous. You would put signs up, and if once you got over 15,000 cells of a species that was toxic, you would be required then to do some testing or analytical testing to determine the toxicity of it. Since then, they realised that different cyanobacteria species are obviously producing different biotoxins and different levels of toxicity, and they are moving to a methodology that looks at specific species, specific cell counts for each of the different levels. And you can sort of see, to deal with a complex issue, they're handling it in a complex manner, and it's not necessarily probably the ideal way of going about it. Current harmful algal bloom management requires expertise for identifying the species, alert level dependent on species, as in the case in Australia, but I assume here too, where you're either looking at an anatoxin producer or a saxitoxin producer. Not all species are producing toxins, and in fact, even within a species, using the genetic test, you find that actually they're only a proportion of what you identify as the toxin species. So sometimes you may be overestimating the potential, or in some cases underestimating. And the dynamics of the bloom alter rapidly, and toxic levels can increase quickly because of sampling events. And in all of this argument, it's the sampling process that offers the largest degree of error, and it's something that needs standardization across all the districts and all the sampling events. So looking, genetics, in a sense, offers a great opportunity to standardize or to look at these things. And specifically, the 3 orders of cyanobacteria each produce a particular biotoxin that's represented. So these biotoxin pathways are obviously cover different species, different orders, and are represented in each of those orders. So a genetic approach, again, if you can target the target pathway rather than just a species ID, is actually probably ideal. And if you look at specialized metabolites biosynthesis, in a general sense, you're talking about sort of a whole group of enzymes within that modules, whether or not they are polyketide synthetase genes or non-ribosomal peptide synthetases. And within these modules, which have catalytic centers that facilitate these reactions and this formation of the amino acids, there are domains that have these centers, and they facilitate a very distinct reaction and the metabolism of a key part of the peptide. So the pathways you're looking for are very complex. Microcystin is at least 55,000 base pairs long. And you're looking for these sets of PKSs, non-ribosomal peptide synthetases, in some form of combination that would allow you to focus in on a biosynthetic or non-ribosomal peptide pathways, and then ultimately looking for the pathway that is producing the biotoxin. And a lot of the biotoxin research and characterization of that pathway was done at the University of New South Wales with Brett Neilan's lab. In the early 2000s, microcystin pathway was detailed. Nodularum in 2004, Cylindrosmopsin in 2008, and then more recently, 2008, for the cyanobacteria saxitoxin pathway, and more recently in 2011, the dinoflagellate marine PSP pathway was outlined. And so these pathways now have a history of about 15 years, and we're still working up these genetic tests to be put into some sort of utility. So microcystin biosynthesis pathway is a series of MCY-type enzymes, PKSs and non-ribosomal. And I suppose in essence you can see that the biotoxin or the toxin is synthesized and ultimately cyclized with the outer compound, and MCYL facilitates that metabolism, but the ADA side chain is in fact generated out of the microcystin E initiation pathway, and by targeting MCYE, you are extensively testing for an ADA side chain formation. And this has an advantage of also being sensitive to nodularum. Yes?
I don't know, I think maybe those are questions, but I guess. No, never mind. Sorry.
We've just got something coming up the screen that might mean a question, so, okay. So that's sort of extensively the specificity, and when in determining what target you're... Oh, okay. Sorry, keep going.
Yes.
In determining your target sequence within all of this, one has to sort of look at the specificity, sensitivity of what you're actually targeting, but also the fact is you're trying to have a broad-based detection system for all the microsystem producers in all the orders. And so a lot of validation needs to go into determine that obviously is sensitive to a number of species. And in saying so, the different microsystem producers, whether or not it's Organosa, an Anabina species, plant threads, you'll find that these enzymes or these domains are actually resorted in different sequences, and so it's not intuitive that when you find these that they are in the order that you would predict in any other species. More so as the biosynthesis, you need to choose the right target, as I mentioned. So we, microcystin E being a fairly specific ADA PKS side chain gene, but there's variations that are induced by the presence and absence of tailoring enzyme. As I said, the MCYL is the synthesis of the ADA to the cyclic chain. But then if you change MCYB1 leucine and MCYC arginine, and they are in different metabolitic states, you actually end up with the LR, RR, or YR. So if you use the specific marker, say in MCYB, you may actually be just detecting a specific conjugate metabolism. So again, choosing the right target actually has an imperative. So with our specific primer for MCYE, we've gone through a number of primary culture stocks around the world with certain references, look to ensure that the MCYE is actually positive in those groups, so that we actually are generating a pan-MCYE target rather than a species-specific. Also, nodularum, again looking at different species across and ensuring that the MCYE target is actually sensitive to those species. And so we have a target that we feel allows that level of sensitivity and specificity, albeit though that there's probably something out there that will throw up something unusual. Saxitoxin, I'm not going to go into the specific pathways, but what's to say is that the saxitoxin A through to, in actual fact, there's an X gene, but again, the different domains can be reassorted in a number of ways within different species, but saxitoxin is initiated by the saxitoxin A region. And so if that region doesn't exist, then you can't initiate the process of production of saxitoxin. And this is in cyanobacteria, but the saxitoxin A gene in the marine dinoflagellate has the same function, but the sequences and the pathway is different. And so when you're looking at Cylindrus mamopsis or an Anabina saxitoxin producer, you can see that the different genes are actually reassorted in any physical presence along the pathway, but they function the same way. And looking at different, again, the target, saxitoxin A for a target, just looking at some Anabinas and Cylindrus mamopsis, just at those producers, the PSC producers, and the target is positive in those as well. So now looking at from a functional standpoint, it's a biological process. If the gene is present, then toxin technically can be produced. So from gene to target. So conversely, if we don't have the gene, you would think that the toxin can't be produced. And so under that premise and, all the interest in the genomics, it was sort of motivation to go ahead and try and develop and commercialize an assay for use and standardization. And then so the first application of this, I mentioned the Warragamba Dam experience in 2007. So this research, well, at least with Microsystem, goes back 5-6 years before this. And when this occurred, while the standard approach was cell counts and then analytical approach for detection of toxin, someone within the Sydney Water Authority handed the lab at the university a sample because they knew of this research and sort of asked to do a series of testing to sort of determine its application. And there were qPCR results done at the university, Sydney Water, and then the University of New South Wales Microbiology Department looking just at the general population, cell population versus the 16S ribosomal RNA gene. And there was consistency, overall consistency in the relative numbers, so that looked good. And then ultimately looking at percent toxic cells versus the actual number of toxic gene cells of MCYE, you can see that while the qPCR results showed an elevation, the microscopy and cell identification and counting also showed an elevation, and then in overall percent toxic genes over the cyanobacteria increased. But the overall percentage of that toxin species or toxin pathway number was only about 5% of the total microsystem load that was present, that whole bloom that you saw in the picture. And it was predicted in that sense that that wasn't sufficient to actually push the toxin level above the threshold. And extensively by HPLC, the actual amount was 1.3 micrograms per litre. And Australia's cutoff is 1.6, just as much as the EPA is here. So it panned out in this case that even though this was not used in the management, this was the first time, at least to my knowledge, that a gene was used in an active management process, albeit not from a regulatory standpoint. So a genetic approach might offer broad level sensitivity and specificity and independence of cell characterization. It can assess the homogeneity or heterogeneity of cyanobacteria population. Not all cells are toxic producers. Not all blooms are individual species driven. Potential for earlier intervention, obviously with a prediction. Discriminate or validate follow-up tests, possible conjugates, species not identified. So if you had a lot of toxic genes but you weren't getting, the ELISA assay or a particular HPLC assay, it might be nodularum, it might be a conjugate that doesn't actually, you're not sensitive to for your testing. Measures the toxin pathway, so that's independent of cell count, and actual might be a better measure for toxin production. And monitors the toxin and non-toxic cyanobacteria population dynamics. So does that percentage of pathways within a total bloom change, and is that change then relevant? So there's been a number of papers over the last 5 years, but in essence probably smaller than I would have thought, given the history of the pathways and the interest in biotoxins. Mostly there are a number in Europe, Spain and Portugal have done a fair. In the Bay Area, there was one in 2010, Baxter et al., looking at trends and sources of microsystem by qPCR. Prediction strategies designed to manage adverse effects. There were, Canada has done a lot using qPCR. And in general, they've all been their conclusions have been supportive of the concept and have shown positive relationships between genes and prediction and toxin. This year there's been probably 3 papers of note. One's looked at environmental influence of cyanobacteria abundance and toxin production in shallow temperate lake. And this paper showed that by measuring the gene, it was more sensitive than cell count, or more to where the gene was picking up things that the cell count wasn't. There was Tim Otten out of Oregon. They looked at the Klamath River and showed that they could detect microcystin E genes and could map it down the river from the outflows of the Klamath Lake. And that was a very good environmental study of and how to monitor and measure the toxin gene down a riverway. And more recently, there was a one that didn't show an appropriate indicator for microcystin concentration by Beaverstoff, Lucas, and Todd Miller out of Wisconsin, and that was just more recently. So within this, there are still one or two papers that don't show that correlation, but for most part, there shows a reasonable indication of benefit. So before I sort of go on the design of the assay, I'll take a step back, and for those who are very familiar with qPCR or PCR, please excuse me, but it is probably worthwhile just when it comes to outlining how the multiplex works. So general PCR is, again, I would assume that most of us understand looking at a target sequence, and that target sequence is probably only about 100 base pairs in length, maybe 200. And when you separate these by heating it, you introduce a primer, which are a complementary piece of DNA, both at the 5', 3' end, and by then, with a Taq polymerase enzyme and all the DMPs, all the base pairs that are used to, produce the extension, the polymerase allows this to extend, and you end up with 2 copies when you first started with 1. And of course, then you cycle and cycle. You're expecting exponential growth in your target, so that there is, and PCR efficiency is denoted by the lack of that logarithmic or exponential growth. So with qPCR, you're adding an additional variable. You're adding a probe into a general PCR reaction, and these probes are generally either beacons or TACMAN probes, dual label probes. There are other types, but these, for most part, are the ones that are used generally. And then this is that you've got your 2 primers in your general PCR, but before you start annealing or extending that sequence to produce 2, your probe finds a complementary piece of DNA in between, somewhere in between the target piece, and binds to that. And this reporter gene and this the reporter fluorophore and quelcher, when they are put together, and fluorescence or light is then put into the reaction tube, the fluorescence from this is actually quelched by this, but as it extends, there is a degradation of that probe. The reporter and quelcher are broken apart, and then when there's a fluorescent on the reporter, an indicative or a resulting fluorophore at a slightly different wavelength is then able to be recorded because it's no longer quelched. And I actually have a little cartoon that probably sort of helps identify this. So you separate your target sequence, a probe finds its appropriate spot. You have your probe or your primer. If you excite the reporter at this point, the quelching occurs because of the close proximity of the quelcher, and it absorbs that resulting fluorophore. But if, on extension, the reporter then gets dissociated, then the resulting excitation, it allows then the fluorophore to be read through the detectors. And that fluorophore is then an expected wavelength because you know what the wavelength of that reporter gene is. So in that sort of sense, when you're doing a qPCR, you're running down a baseline where the overall concentration of the genes you're targeting are not measurable. It's only until a certain time point that there is enough PCR product where the excitation of the reporter will show a meaningful reading of the excitation. And at that point, it will start this linear phase, which is the exponential rise in your product. And the closer that is to a linear phase, the better the efficiency. And then you run out of reagents or the plateau stage. But what you do is you measure the CT value, the threshold, where the overall fluorescence is above background or above any non-specific reactivity. So there's your curve, but in a multiplex assay, because you're now looking at a system that can report based on a wavelength, you can introduce different colored probes with different wavelengths, and if your detector can detect those different colors, you can actually have multiple reactions going on. And in this case, we're having 4 colours and 4 unknowns can be measured. And there are multiple pieces of equipment out there that can perform these reactions, as you probably all have one somewhere sitting in the lab. And in this sort of sense, in 2011, we published a paper putting a multiplex qPCR together, targeting Cylindro, Microcystin nodularum, and a Saxitoxin. And a lot of those papers that have been published are mostly single-target type assays, and this was really the first multiplex looking at a multiple biotoxin pathways. Now, this is a collaboration with Brett Neilan's lab in my company, Diagnostic Technology, and very importantly, the National Measurement Institute, which is part of the Australian Government's Department of Industry. And I asked them to develop some genetic standards. And the principal reason for that is that, given that this is a highly regulated environment, and if it was very important to standardize a target or offer some standardization, that when testing occurred, there would be that standardization could be compared to different regions, different countries, and different river systems. And the National Measurement Institute is actually part of an affiliation of standardized laboratories around the world, Standards America included, the EU, and other bodies. And so they are actually have the authority or the ability to certify standards and to produce things under ISO manufacturing. So they were put to task to produce a homogeneous, stable sample with specified properties. It had to be fit for purpose, fit for this application. It's an authority body, so that they're independent and qualified to do this. They're in no connection to us, at least commercially. Give numbers, traceability, and give confidence levels with uncertainty. The process is not any different than probably most researchers take when they actually develop a set of standards in their research, which is to do a construct of the DNA sequence that you're actually measuring. But in this case, we produced a single plasmid construct that had each of the targets contained in the assay. So a 16S target, a microcystin, and a saxitoxin target from the publication. And the 2 methods, and you actually then have to do 2 methods of quantitation to ensure that you've got accuracy. They used isotope dilution mass spectrometry and digital PCR. And digital PCR, of course, is actually now regarded as being sort of a method of choice in some labs, because it doesn't require standards or standardization. It's an absolute quantitation type system. But the systems are more expensive and do require higher cost reagents. So this is just a graph showing you that there's a one-to-one relationship for each of the targets. So the plasmid would generate full-length DNA that was measurable at a one-to-one ratio. It would be no good if there was a preferential amplification of one target over the other, because these targets are contained in a single vial of standards. And looking at the replicates, we're dealing with a percent CV of around 4%, 1%, or 2%, 1%, 1%. And here's sort of a graphical representation of the standards run through a qPCR, looking at the standard curves. And these are highly reproducible and allow for, in one assay, sort of identification and standardization. This is then the stability. We've run them, well, we've actually run it over 12 months now, and the stability is excellent. We've done transport stability at 22 degrees up to 14 days, and minus 20 degree storage now over 12 months, 18 months, and it's all looking as we would expect it to do. They've assigned standards to this. In fact, so we have a 20 or a 19 per microliter. In the assay, you use 5 microliters, so this extensively is 100, 1,000, 10,000, and 100,000. And for reasons of practicality, we're now adding a 1 million copy standard to the assay to cover some applications. So the specifics are is that we simultaneously identify and quantitate the presence of cyanobacteria from 3 genes responsible for 4 different toxin production pathways. We do a specific Cyanobacteria 16S ribosomal RNA gene to give an overall Cyanobacteria load. It also acts as a control, because if obviously you have Cyanobacteria in the water, you would want that to be positive. We also include an internal amplification control to ensure that the assay is functioning and there's no inhibition. And then in a separate multiplex, we do a microcystin nodularm using the MCYE gene, and a cylindro and a saxitoxin. And the test in itself in the PCR is only an hour long, unless you're using an ABI system, which generally takes a couple of hours. But it's scalable, as in you can do as least as 2 or 3, 4, and do as many as you like. It's cost effective, and it targets the pathway, not the species. So in a very sort of general methodology explanation, you prepare the sample, which is where a lot of the variation occurs, but trying to simplify that. But you prepare a sample, you rehydrolyze the bead enzyme mix, and if any of you have used the BioGX technology, it's actually BioGX who I work with in manufacturing this test. So you rehydrolyze, this contains all the probe primers, DMP, magnesium chloride, and the Taq polymerase. You add 5 microliters of this prepared sample, and you run the PCR. You would have already pre-run the standard curve, and then your results are then equated against that standard curve. So kit comes in 2 pouches, 2 separate assays. One for the Cyanobacteria 16S with the AMP control, and this quantitates the total Cyanobacteria and determines if a dilution is needed if you want to have absolute quantitation. But also in a screening type situation where you have very little cyanos, you may not necessarily need to go to the toxin gene assay if there's not already a threshold of 16S there to equate any level of risk. But should you run the toxic gene assay, you could do that parallel or in sequence. There's the target genes for the different pathways, and it quantitates that. So in trying to summarize what you may get out of this approach, you'll be able to say, I definitely got Cyanobacteria. It indicates the measurable level of that Cyanobacteria. Do I have toxic gene presence or a species that has a toxic pathway, yes or no, at least for those 4 toxic pathways. It'll give you actual amount of that. You can actually relate this to the gene load or the total Cyanobacteria content. And in that way, you have sort of a number of, interpretation criteria that you can work on. So this is just one sort of a study, a poster presentation at one of our Cyanobacteria meetings. It extensively shows sort of a city-based lake system in one of our parks. It drains into a large bay near Sydney, and they generally have some level of microcystin toxin production during the summer months. And this is just an example which has been highlighted in any number of those papers that I referenced before. But if you see your toxin quantities increasing, you also see some sort of masking of then the gene number increasing as well. So this was a study in 2010 looking at the Murray River catchment, looking at sort of the incidence of the different toxin, the pathways, or the presence of different species in one of our largest river systems. And 60 samples were tested over 1 month in 2009. The PCR assay was used. And you can sort of see the assay was able to sort of look at different sample sites, different dates, and actually tell you if all 3 pathways are present in the sample or not, or only one, or what combination. So it had the versatility in one sample to give you some heads up in a number of ways. Now, I in the last few months, I've had the opportunity to run the test in Ohio, and I could show you a couple of sort of data points that I think are interesting, and show some sort of indication of some utility. This was Lake Rockwell. This is the water intake for Akron, Ohio. It's a fairly small reservoir per se, but it's the intake is down here at the dam, and the water treatment plant is down here. They normally take samples at the top, the mid, and the dam intake, and this area is shallow. It has a number of tributaries in here that have some population or some homes and other activities here, but this most part is natural. So over from July, when we first ran the assay, at the top of the waterway, we were actually getting 2,000 microcystin toxic genes, and the midpoint in the dam gave nothing. And this reservoir lake has a 2 to 3 week retention time, circulation time. And then subsequently, the top was still showing some signs of microcystin or gene pathway, but the mid and the dam suddenly started showing up. And then subsequently, a month later, the dam was still positive. And when I show you the next slide, 10,000 gene copies is probably not something to get too excited about. You would you'd like to sort of monitor that, but when I show you some of the other levels that were showing in Ohio, you'll get a sense of this is easily managed. But this was sort of one example of they always suspected that the top component was where the source came from, and that sort of bore that out. And they were able to, I suppose, to get some idea that, if that was traditionally how it happened, by monitoring that, they would actually get a heads up what's going to happen, sort of thing. But this is, again, only one sequence of events over one season. But they actually had a very large bloom towards the end of the bloom season. They picked up a small number of saxitoxin, which is not necessarily monitored in Ohio, at least this last year, but it will be for the years to come. But there were no toxic gene pathways in that, and that was reassuring to them given that it was a very large bloom. So these were two reservoirs, or actually a number of samples. So the first one is a sample that I got from the Great Lakes Environmental Reference Lab up at NOAA in Ann Arbor, and the 16S was up around 6 million. The microcystin gene contact for Lake Erie in July was 300,000 gene copies. That sort of shows that about a 6% content of the bloom that was happening in Lake Erie in July had a toxic pathway. And I don't have the representative ELISA results of the toxin. So these beaver and raccoon and Patmos and harsher are reservoirs within Ohio. I only have sort of 2 samples for beaver over a period of time, but looking at just the percentage, you actually had up to 12 or 10% of the overall bloom, which was well over 1 million, half a million, and that reached a peak of 170 micrograms per litre of microcystin. Subsequently, if you look at Patras, where there was only 4%, 5% of about 1 million copies, you only had 28 micrograms, so slightly above the recreational limit, but well managed at the intake level, and there was no recorded levels of microcystin that were outside the limits. So in some sense, you've got some sort of feeling that high toxin levels will tend to be a large proportion of the cyanos overall, and actually have a fairly significant level as well. So this was then the microcystin by ELISA over a period of the summer, and the X's are where we got an opportunity to test. So we tested them at times where the toxin levels were at its peak. So you would hope to think that that's obviously the gene would be present, but what would happen down here, I can't say. So this is Barberton Reservoir in Ohio. It's a long, thin reservoir. And they actually had some saxitoxin ELISA positive, and they weren't able to detect any specific cells that were responsible potentially for saxitoxin. So taxonomically, there was no actual indication at this time point. They then sent some samples to be analysed. We got anywhere between 1.5 million and 400,000 16S, but there were small levels of saxitoxin genes, so at least it aligned with the presence of saxitoxin. And we subsequently, then they thought, well, let's go and sample around that reservoir to find its source, because it could be benthic or otherwise. And lo and behold, on the west bank and on the boat ramp, at least in August, because this was a week later, there were larger levels of gene, and that sort of gave them an indication of where that might actually be occurring. And then maybe in subsequent management or subsequent years, that might give them some indication of how to manage that area. In other areas, it was small. So, and these particular samples, looking at the whole timeline of saxitoxin ELISA tests, were right at the peak. So again, sort of fitted in with the microsystem. So Ohio River was closed for a number of times. They had a 500-mile algal bloom, and the microsystem levels were up to 630 micrograms per mil. And that was being, at least one sample I ran a couple of weeks ago was up to 300,000 microcystin genes within that bloom. So, correlation. But the Ohio River was actually running sort of fairly hot. So just last week, there was a publication by USGS out of the Columbus laboratory, Donna Fracey and Darren Seltzer. And this was just published last week. And basically, long-term prediction, the most highly correlated molecular assays were the MSOE Planthrix and the Buck Island. They actually monitored a number of reservoirs and beaches and looking at the microcystin E. So they actually did species-specific or family-specific microcystin E, not a pan microcystin E, looking at what may be the best sort of target. So these were the study sites. Buckeye, Deer, Buckeye, and this was the East Fork of Harsha. And while this slide's this is a direct, I've pulled this straight out of the publication, but if you look at sort of the summary statistics and looking at the water quality measurements, you would see that the microcystis or the microcystin E DNA was 88% positive, 90 and 100% in those dams with the presence of cyanobacteria. This is a relationship between the gene concentration and the community compositions, and there was a general trend between the genes and the cell number, which you would hope to think. And these are then the relationship between environmental and water quality factors with microcystin concentration. So here's the gene number versus the toxin level. And not all directly, good correlation, but compared to some of the other factors, highly correlative. And in the sense of this is the water quality environment factors on a daily long-term prediction, and this is from the highest correlative test to the least. And in three of the dams reservoir, you had cyanobacteria qPCR, and then the microcystin E, and So microcystin E was the best in Harsha, and came in about 4, that's still 0.82 for Miami, and Buckeye was at 0.51. So it's still variable in those situations, but it still overall had a very good long-term predictive value. And coming to an end, I just quickly a few slides. We've since now outlined the pathway for the marine saxitoxin. An assay was put together, and we're currently doing some validation across multiple species, saxitoxin-producing species in dinoflagellate, and we're showing good sensitivity, specificity, and hopefully shortly for the marine side of things, we'll have a quantitation assay for PSP identification in shellfish, or waters for shellfish. Other than that, I'd very much like to show my appreciation to Ohio EPA, particularly Nick at the lab in Columbus, the lab manager, and Heather Raymond. City of Akron with Jessica, who we've worked with for the last couple of months, just getting some of that data, and Northeastern Ohio Regional Sewerage District with Mark and Nikki. But other than that, the people that have been involved in the development of the assay. So thank you, and I'm assuming I can take questions.
Does anybody out there have questions? Star 6 and ask away.
This is Akin Babatella, City of Santa Cruz. I don't know if you can hear me.
Can hear you easily.
Okay. It shows that I've been disconnected for quite a while, so I didn't see most of your, I suspect, the last 6 to 7 slides.
Okay.
But I could hear you. I had asked a question about correlating the DNA copies to actual toxin levels. And I don't know if you showed anything related to that. I sent the question via chat much earlier.
Okay. The room we have here is the actual computers down the back, so sorry if we missed that question. There is some papers that show good correlation between the gene copy number and toxin, and some different river systems, and some papers that show poor correlation with. Some have explained that more frequent testing probably is required to determine that, and others have suggested, either a delay or otherwise. But I suspect there is the variation in sampling has a lot to do with it, and standardise the sampling within a waterway, or knowing how your specific environment is probably essential in this. But the data that, or at least the situation we found in Akron, was that while we detected a small number of microcysteines somewhere, it obviously over a period of time extended out. But if a bloom collapses at some point and releases toxin, as in the saxitoxin experience in Barberton, where the saxitoxin they were detecting in the water was all extracellular. There was very little indication, and the gene assay picked up the genes, but still there was not a great deal of actual whole species that they could identify with. So I think I'd only feel comfortable with this one is that there is some level of correlation. It's predictive because obviously you're not going to get any toxin level. The single time point assays that have been performed have always shown that there is a reasonably high number of gene copies in samples where there's high toxin levels. But I think more timeline studies need to sort of occur to get a very good understanding of whether or not this proceeds by a month, a week, or two, or what happens.
Thank you very much. That was very helpful.
I think I may be it. Is there any other questions out there?
Yes, this is Marissa. Can you hear me all right?
Yes.
All right. Thank you for the presentation. I was hoping if you could just scroll back to the slide a little while before you got to the examples, describing what kind of data is obtained by each assay. You know how you had first started the slide off with you learn if there's a presence of cyanobacteria.
That one?
Yes.
So, yes.
Okay, so I think I might have missed this out later on, but what's the units for the amount of toxin? Are you just getting gene copies, or are you able to correlate that back to a milliliter?
No, I wouldn't want to say that at this point. Not enough data to support any interpretation of that. Mm-hmm. There's been some publications, and please also understand that a microcystis originosa may actually be more productive than an anabina at producing microcystin, or a different level for saxitoxin. So it would be very hard to generalize sort of the amount of toxin produced by any different species or different pathway. Yeah. So some have sort of suggested there's about 1 femtomole toxin per cell, but is that per day? Is a bound when the cell ruptures? How much is released? So the assay, from an analytical standpoint, tells you with a high level of standardization how many genes per mil there is in the sample that you have collected.
Okay.
So
thank you. That totally answered my question. Thanks for giving some background on that.
Thank you.
Okay, is there anybody else? Okay.
Thank you very much.